Good morning, everyone. Thank you for listening. Today, I'm going to be talking about cartridge samples and just a little bit of information about me. I'm Kari Danser, like I said, and I've been a forensic analyst at cybergenetics for about a year. And in the meantime, I've been focusing a lot of my time and attention on this cartridge study. So for some background information, cartridge casings are the empty shells left behind after a gun was fired. And they're typically of different metals and different calibers, so they're different sizes. Nearly 200,000 cartridge cases are recovered annually at United States crime scenes. And these can range from homicides, aggravated assaults, robberies, gang-related crimes. They can really happen with anything with a firearm. And in my time at cybergenetics, I've personally dealt with 2 cases that had cartridge casings. And as a company, we have analyzed over 90 cases with cartridge evidence. So it is a common piece at crime scenes. And it is shown that cartridges that were fired degrade any DNA that was left and have significantly less DNA, which makes it harder for crime labs to interpret the data and even crime scene investigators to collect any DNA from those cartridge casings. And it was also shown that the caliber of the firearm did not have any impact on the amount of the DNA recovered, so it was not a linear relationship. So you can't say like the bigger the number, the more DNA that was left. While I was doing research on the cartridge issue, I came up with 4 main questions that I really wanted to answer with this cartridge study. And one of the questions is, can manual interpretation obtain DNA information from cartridge data? That is the typical DNA interpretation method that most crime labs use. So then I wanted to introduce TrueAllele casework, which is a probabilistic genotyping software. So can TrueAllele casework interpretation obtain DNA information from the cartridge data? Which collection method is the most informative from cartridge data, as there are various ways to collect DNA from cartridge casings? And since there are many various cartridge types, which one are you more likely to get DNA from? Which led me to my study design. This study was broken down into 6 main steps. And the first step was sample creation. And then it led us to DNA collection and DNA extraction, as well as manual interpretation. And George Washington University handled the first 4 steps. They did the sample creation, the collection, extraction, and even the manual interpretation for us. So once they produced those electronic DNA data files, they sent it over to cybergenetics for TrueAllele interpretation. So once TrueAllele did its calculations, we were able to look at the results and then compare the results from TrueAllele's interpretation to manual interpretation to see which one was more informative. So our first step was sample creation, and ideally we wanted to do single source data, which meant that one person touched the cartridges, and that was the only expected person on these samples. So the reference individual touched 7 different cartridge types for us, and we focused on fired and unfired. So for fired, the individual would touch the cartridge after it was cleaned, load it into the firearm, and then shoot the firearm. And we really looked at 45 fired and brass fired. And then for the unfired samples, the individual would again touch the cartridge after it was cleaned, load it into the firearm, and then remove the cartridge. And there were 910 total cartridge casing samples, and we can see that in the table where, for instance, we looked at 45 fired with 90 total cartridge casings of 45 fired. So our next step was DNA collection, and we used 5 collection types. So the first one we focused on was wet collection, and to go into a little bit for each method, a sterile wet cotton swab was taken and applied with some force and rotated on the cartridge for the area of interest. And then it was followed up by a second sterile wet cotton swab over the same area. And then for the wet dry method, sterile wet cotton swab was rotated with force on the area of interest, followed by a dry sterile cotton swab to collect any genetic material. And then we used the soak and sonicate method, where the cartridge was placed in a test tube with some buffer solution, and it was placed in a sonicator for some length of time. And after that length of time, the cartridge was removed from the test tube and the buffer solution was sent on for processing. We also looked at tape lift, and it's where 2 sterile tape dots were placed on a microscope slide, and the cartridge was rotated over those tape lifts to collect any genetic material. And then scraping, where a sterile razor blade was used to gently scrape off any visible genetic material on the cartridges. And the table below just shows the total number of cartridges that were used for each collection method. And if you see NA available for 45 fired and unfired, or NA for not available. Those methods were not used for 45 fired and unfired, so there is no data for those methods. And if you look at soak and sonicate 2 for brass fired, there was only 10 samples collected of soak and sonicate. The next phase was DNA extraction. And the George Washington University lab decided to use organic extraction for this initial phase. And that's where organic solvents are used for denaturation. And then those denatured proteins are removed, then washed. The sequencer that they used at the lab was the Applied Biosystems 3500 genetic analyzer. And the STR kit was the Applied Biosystem Globofiler kit, which targets 24 key STR locations. And at this time, too, the reference was collected and extracted as well using the Globofiler kit. So once all the data was generated, George Washington University manually interpreted the data. And they applied a peak height threshold to the EPG data to form allele events. And I like to use this skyscraper image as an analogy for thresholds. This was from my coworker Stephanie, as she taught me this, and I think it's amazing. So basically you have a city, like Pittsburgh outside or even New York City, and you see like the tall buildings like the skyscrapers, you see the little buildings like apartments and shops, and then you see the tiny buildings like houses and even like garages and things all throughout the city. So if you were to go into a building and go up to the 20th floor and draw a horizontal line across the city, like we see here in the image, that would be your threshold. So you could only look at peaks above the threshold. So you would only count like the tall skyscrapers, like the two with the pointy satellite things on top. Those would be 2 peaks that you could count. But you lose a lot of information as you're discounting a lot of the smaller buildings. Like you're discounting any building that's below the 20th floor. So you're losing a lot of DNA information there. And this is really one of the limitations of manual interpretation. So for this study, they focused on allele counts, which is how many EPG allele events matched our reference of interest. So then once manual interpretation was completed and they sent over their results, they also sent TrueAllele the electronic data files. But before I go into the TrueAllele process, I just briefly want to explain TrueAllele. And TrueAllele will look at the proposed patterns of the data to determine genetic types. So basically, this is just a nice explanation of how it uses colored boxes visually for us to explain the data to match up the underlying data peaks that we can see in the red. And we see the boxes line up at the top of the peaks, so this would be a good explanation and it would be giving a higher likelihood. So then we could take those explanations that have a higher likelihood and give them some sort of probability. It's how many times did the computer propose this was a good pattern. And we can see that in the image on the bottom right. And we can see that for this example, the reference's allele pair is in a red box. It's the 2425. So we can plug that into our little formula, which is the probability of the evidence match, or the blue bars, over the probability of the coincidental match, which is how likely is this allele pair in the population, to get our match statistic for this location. So if we do that real fast, we take the 30 over the 4, it's roughly 8. So it's 8 times more probable than a coincidence for this location. So we do that for every single location, multiply them all together to get the overall match statistic. So now that I explained that, we're going to look into what we did for this study. So the cybergenetics trained TrueAllele analyst generated single source requests. So basically, he asked the computer to solve for one contributor. The computer is completely objective and unbiased, so TrueAllele processed the DNA data without even knowing a reference. So basically, we set up all of the single source requests, which took about a week and a half. And once all the single source requests were run, we put in the reference into the computer and asked it to solve for the reference. The computer also generates the Legler genotype statistic, and this quantifies the identification information in a genotype, so it shows how informative that genotype is. And it's the expected log LR to the true contributor if it's unknown. The computer also calculates a likelihood ratio match statistic, and this compares the genotype to a known reference to get the answer that we're looking for. Is this person present? Is this person not? So for our final information comparison for part 1 of this study, we reviewed the KL information first from all of the single source runs. And we can see that most cartridges had a high KL on the table below, indicating that the DNA was informative. And keep in mind, this is the number of zeros after a 1. So you may see that it's a 4, but a 4 is actually fairly informative. It's not the highest, but it shows that all of this data is informative. However, there was a problem upon review of the data. a lot of the data was low-level mixtures, something that manual interpretation didn't even consider with this study. However, we were able to consider and revamp the study because mixtures in low level, oh no, what do we do? So upon review, we noticed that a lot of the locus peaks, or locus EPGs had 3 or more peaks, which indicated that 1 or more person was present on these cartridge samples. The data was also low level, which meant there was little DNA, and oftentimes it was uninformative for manual interpretation. So this table shows the percentage of the low-level samples for each cartridge type. And if you can see, aluminum unfired actually had 0 low-level samples, which was pretty good. But the others had high percentages, meaning that a lot of the samples were low-level. And it was interesting because manual review couldn't handle more contributors and sub-threshold peaks, so a lot of these samples would be considered uninformative for the study. So now that we looked at the low-level data, we also looked at mixture data. And TrueAllele specializes in mixtures, so TrueAllele was to the rescue to save the study, so I didn't have to revamp it. 431 of 910 samples were found to be mixtures, which is 47% of the samples. That's a lot, it's almost half. And allele counting couldn't handle more than 1 contributor, considering it was only looking for the references allele pairs. And this table just shows the number of total mixtures for each sample and collection method. Again, there was no data for 45 fired and unfired for tape lift and scraping, but we can even see that the brass unfired for silk and sonicate did not have any mixtures. So manual interpretation could have given us an answer for that, but explained the other samples. So for TrueAllele interpretation round 2, a trained cybergenetics analyst created requests for the mixture data, and the TrueAllele computer processed the request. But what was interesting is, for our casework process, we look at the data, and this was done just like our casework process, to determine the number of contributors. So we looked at the peaks, and we counted them to see if there was like 3 or 4 contributors. Well, some of the samples were undetermined. It could be 3, it could be 4. So we ran it as both contributor assumptions and counted both for our study. And these samples contain either 2 to 5 contributors, with 5 being the max. And TrueAllele found an unknown person in many of the cartridge samples, so then we were able to compare the cartridge samples with this unknown profile. So now that those requests were solved, we can look at the information comparison again, now with mixtures and low-level data. So all cartridge samples were compared to a reference, and we can look at this table to see the number of inclusionary counts for the reference, which we expect this to be higher since we wanted this known person to be on the data. But we see for like 45 fired, nickel unfired, and brass unfired that there were no inclusionary statistics for the known reference. So then we did this for the unknown profile as well. We counted the number of times that the unknown profile generated an inclusionary statistic. And this was honestly shocking. I was not expecting this unknown person to be across multiple different cartridge types. And there were even 4 instances where the unknown person was not present. So the unknown profile was in 138 of the 910 samples. So that would have been 138 samples that manual interpretation only focused on the reference and discarded this useful DNA information. The unknown profile was quite informative. Its KL was 30.36 ban, which means it's 30 zeros after the 1. So that is quite a big number. And this profile was also single source. So whenever we looked at the data, we found that this single source profile was very high, but it did not match our reference. So we then took the genotype from that profile and compared it to the others to get statistics. So this study was a lot. There were 31 different tables of the collection and material type. And to be honest, I could have sat here and went through all 31 of those, but I didn't want to do that, and I didn't want to make you guys sit through that. So I only focused on one, and it's the nickel unfired wet collection. And this is the table of the statistics for the inclusion for the unknown and the reference. And a blank entry indicates that there's no data available. So for this, there were no single source requests that had an inclusionary statistic to the unknown. And this shows the KL and log LR values, which are the number of zeros after the 1 in the match statistic. And the inclusionary LR values range from tens of billions to tens of quadrillions, which shows that this data is really, really informative data. So that means we were able to get data and information off of the cartridge samples. So I currently have 3 interns at the office, and I make them do presentations, and this slide was really inspired by them. So I decided that it'd be a great idea to see how many zeros after the one that a 10-man is written out. That's a large number. It's a lot of zeros. And it's a large inclusionary DNA match statistic. And keep in mind too, this is the average from a 3-person mixture. So that means we got this high statistic from a mixture with 3 people. So TrueAllele was able to separate out those genetic types and make a comparison and say that this person was in there. However, human review did not get this information at all, considering they only looked at one person's allele pair, so they expected one person to be there. Now that I got through the boring math, you're probably like, well, who is the unknown? I don't know. That's a great question. I'd like to know. We believe it's someone who handled the gun. I'm not sure if the gun was cleaned before analysis, but it was interesting because it was the same person across multiple cartridge types, and it was not restricted to a specific type, which to me indicates that it was not somebody that manufactured the cartridge. Because none of these cartridges, I believe, are manufactured by the same company or even bought at the same store. So that led me to believe, was it somebody that was potentially on the cleaning materials? Was there some sort of contamination there? Eventually, if we ever find the profile for this reference or the identity, we could make a comparison to generate match statistics, but I doubt we ever will. So some overall study conclusions. Manual interpretation used allele counting and thresholds, where they could only find the known reference, but that's all they were looking for. Those thresholds limited the peaks that they could see, and since a lot of the data was low level, they only looked at about half of the data. TrueAllele considered additional mixture contributors, as we were able to go into the computer and look at the data and determine there was more than one person present and separate out those genetic types. And the computer calculated match statistics for both the reference and the unknown profile that manual interpretation did not even find. And the computer developed that profile and compared it between the different cartridge types for us to determine if it was specific to one singular type. And the known reference is found in 351 samples, and it was found in 205 samples manually. And the unknown person was found in 138 samples and 0 manually, as they did not even consider looking for an unknown. For the more informative collection method, we found that wet-wet or wet-dry was the most informative, and tape lift was a close contender. And this was determined by the KLs, whether or not the KLs were high and informative, and the number of inclusionary statistics for each collection method. The least informative was scraping and soak and sonicate. Again, these methods had the lowest KL and lowest inclusionary statistics. The most informative cartridge type was aluminum and steel, and I based it again on the KLs and the inclusionary statistics for each cartridge type, for at least organic extraction method. The least informative was 45 fired, but there could also be a small limitation with the 45 fired sampled, as it was not collected using scraping or tape lift, so that it was a smaller sample size to work with. For an overall conclusion, TrueAllele can develop informative data from cartridges, and all DNA data is used and none is discarded. Like for instance, with the thresholds, TrueAllele does not use a threshold, so we're able to look at all of the data, even the low-level data. And we can handle the low-level data and look at minor contributors and small percentages, like really minor contributors, the manual interpretation cannot look at. Cartridges are common crime scene evidence. As we saw in the beginning, 200,000 cartridge casings a year. It's a lot, and a lot of crimes nowadays involved handguns and any sort of weapon. So this can be a motivation for crime scene investigators and crime labs to gather cartridge evidence, as this is a tool that can be used to gather informative cartridge DNA. Methods that use the less data are less informative. We saw that with manual interpretation, they used a smaller amount of the data and had less inclusions to the reference. And this is interesting too, just a reminder that TrueAllele has done almost 100 cartridge cases and has gotten results. So we were able to get more DNA information from crime lab data, working as a complement with the crime lab to help solve these crimes. For some future directions for this study, this is just phase 1. I'd like to analyze more cartridge data, and we have other extraction methods already prepared and ready to go in the request or solving. So I'd look for PrepFiler and CAIAMP extraction methods on these same cartridge types and collection methods. And I'd like to determine the best collection method for each extraction, and even the best collection method for each cartridge type. Does one collection method and extraction work better for cartridge types? And look at various combinations of that as well. And I would like to be able to compare TrueAllele casework interpretation with other interpretation methods using KL and log LR to see if we can gain more information from other methods, and even if other methods are working as well as TrueAllele. Before I ask if there's any questions, I'd like to acknowledge MAS for letting me talk, the people at Cybergenetics for giving me this opportunity to look at cartridge data and keeping my head on my shoulders when the study went by the wayside with the lovely problem. And I would like to acknowledge George Washington University for their use of their data and equipment to make this study happen. Thank you.